Okay, good morning, everybody. Let me get this full screen. Happy Friday. We will hopefully wrap this up relatively quickly and start chapter 14 here, at least get some of the introductory stuff with chapter 14. So I left off, and also there were questions Wednesday as far as which chapters 13, 14, 17, 18, 19 are what we are covering in this unit of material. So 5 chapters that is posted on the schedule on Moodle as well. Also looking at the schedule, I tried with the second exam, I was like, oh, I should stop this at 5 o'clock. And then I looked at stuff posted online that said things closed at 11:59. So then I switched it back to that. It will be the same. I'm going to leave it at 11:59. So you have a little bit of time to deal with that. So with that, I guess any questions before I get going? Give me a second. Okay. So this, we left off here with the hand washing procedures. This is something that that I know we have thought about in the lab. It was, if I recall, the very first lab that you went through. There was a video with a TA showing, demonstrating some of that stuff, and ultimately, something that I hope, as we're all working through this pandemic, that we are all aware of. So with that, real quickly, when you're doing this, and I don't think I really need to tell you how to wash your hands, this is microbiology, but you want to make sure and wet your hands. It doesn't truly matter if it's warm or cold water. A little bit warm water helps to kind of get the oil laters on the surface of your skin a little more loosened up, if you will, but again, not super critical. So apply soap, lather it up, working it onto the palms, onto the back side of your hand, in between your fingers, the fingernails, trying to ensure that every surface of your hand is thoroughly lathered up. After you've done that, you want to scrub roughly 20 seconds or so. It says sing happy birthday to yourself, maybe twice. That's about 20 seconds. And then rinse. I feel like, 20 seconds, give or take a little bit, as long as you've thoroughly covered the surfaces, you're going to be pretty good. Rinse your hands well with running water, rinse off all of that soap, dry them off, you're good to go. Back in business. The image below is a little more detailed. A surgical scrub in is definitely more extensive in this process. Again, washing your hands thoroughly, but starting from the fingertips, moving all the way up the forearm, a little bit past the elbow, hoping to ensure that as much of the transient microbes, those that are potential pathogens, have been removed. You can't guarantee that every single organism is off of there, but most. And in the process of doing this, you know that microbes are all over the place. I assume when you looked at the hand washing stuff, and we did it a little bit different than some of the labs before, but with this, you probably saw organisms before the soap and water and after. In the past, we did a handful of washings where you initially just wetted the fingers or wetted your hand, touched the plate, washed with a little bit of soap for, a few seconds, again, touching a quadrant on the plate, washing for a couple of minutes, and then it was up to about 5 minutes, trying to kind of replicate a surgical scrub in, which usually is more than that, a little bit more lengthy. Oftentimes, you see a considerable amount of organisms after you've washed for a while. And I feel like it gives the wrong impression. Lots of times people see that and they're like, holy cow, I'll never wash my hands again. Look at all of that gross stuff. Really what you're seeing are your normal flora that's underneath that oil layer. Those are the good guys and you're letting them shine through. You want that stuff to happen. In a surgical scrub in, they're trying to remove as much of that as well, to ensure that, if they are, working on you doing surgery, even though they're gloved up and all of that, the potential for possible contamination is dramatically minimized. I did notice a chat here just a second ago asking when in-person lecture was going to start. That will be Monday. I will be back in Fraser Hall on Monday. I will still be recording stuff like we are usually. I kind of feel like this YouTube works a little bit better for me than recording stuff on Zoom, so I probably will keep posting that way unless there's issues with that in the classroom. But I think that should all be good. So Monday morning, I will be in person. It will be nice to be back in action. Okay, let's see. So again, some more molecules. I don't want you getting bogged down with this, but these are compounds, antiseptic compounds that you see in surgical scrubs. I know that with some techniques, alcohol-based scrub-ins are kind of replacing some of that and minimizing the length of time that's needed. I'm not someone that goes into, scrubs in for surgery or anything like that, so I don't have details about that. But I have been told that there are some different procedures that kind of minimize that a little bit. So some more agents, and again, I'm going to go over or gloss over all of this because truly you don't need to go through the details of all of this, but all of these have the potential of serving as alkylating agents and ultimately preserving or lysing cells. Catalase activity is something that I think you're going to see. I'm trying to recall if you've already seen this. I think you're going to see this pretty soon as we start thinking some of the respective biochemical tests. It's something you're probably a little bit familiar with. Peroxide, typically when put onto something, you might see some bubbles. And if you see that, you likely it's the activity of catalase that's breaking this molecule, the peroxide molecule, into water and oxygen gas. So the bubbles you're seeing are liberated oxygen through this conversion. So there are all sorts of ideas with this here where it looks like something cleaning contact lenses. There are other, solutions for that, but this, I believe, is what we're looking at here. And you can see all of the bubbles within there. So as far as bacteria go, there are all sorts of possibilities with this. And there are also some potentials when we think about some of like the biochemical tests to help us to differentiate and maybe rule out or rule in specific types of microbes. There are numerous types of organisms that do have catalase activity. So you can't just say, oh, this keys me in. But there are a couple that we know that do and a couple that don't that are important in biochemical assays. Specifically the staphylococci. These guys are catalase positive, and the streptococci are catalase negative. So through a gram stain, you might see gram-positive cocci. The staph really should be clustered, and the strep should be in chains, or maybe diplococci, depending upon the species. But sometimes through a gram stain, that's hard to differentiate. There may be a really thick smear where the cells are really just stacked up on one another, and that might be a little bit misleading. So growing up a culture, adding a drop of peroxide onto taking some organism and putting it onto a glass slide, and then a drop of peroxide on top of that. If it bubbles, like you're seeing in this contact solution over here, if you're if you see the bubbling, that indicates that it's a catalase positive organism. If it's not bubbling, it's negative. And again, ruling in or out, staphylococci is really what we think about with that. So it's also important when we think about, potentially using it to clean wounds. You do want to kind of use some caution with that. Excessive use of peroxide can cause tissue damage, but it is important when we think about the potential to kind of clean or cleanse a wound, to de-germ, as the peroxide gets in the bubbling action. And you see this with your red blood cells, there's catalase activity. So you see bubbling occurring just kind of naturally. That helps to kind of flush out those specific organisms. With that said, we may think about using peroxide to, think, oh, I don't want to get a staph infection. Well, the staphylococci see peroxide and they chew it up, basically. They're like, oh, well, I can deal with that. I'll make water and oxygen out of it. So it's not actually targeting or killing the staphylococcus, but it does help to, through that bubbling action, helps to remove those organisms from out of a wound. Other things that do not have the catalase activity definitely are targeted by the peroxide, and it would target them. So that's kind of just a little side. This image is an antimicrobial susceptibility test. It's Kirby-Bauer technique, or disc diffusion. There are all sorts of different possibilities as far as antimicrobial agents go. Usually, and we will in the next chapter go through some more of this. I kind of feel like this image should be in there as well. In the text, it does reference back to this image in chapter 14. It references this right back to this. These, although here it looks like just a normal sized Petri dish, typically they're much larger Petri dishes that allow for numerous antibiotic discs to be placed onto the plate all at the same time. That plate gets incubated, and then dependent upon the size of the zone of inhibition, as we see on these plates, the greater the zone, the more effective that specific antimicrobial agent is. There are, again, numerous types of antibiotics that might be tested. Usually when we do this stuff in the teaching labs, we have the dispenser, although here it's a little misleading showing 4 or 5 discs. The dispenser we have places 12 antibiotics onto the Mueller-Hinton plate. This Mueller-Hinton auger is a specialized type of auger that allows the antibiotic to diffuse evenly into the medium. You could possibly use nutrient auger or other types of media, but the issue with that, some of those formulations with the auger, they don't polymerize the same. They don't, it's not an equal type of polymerization, which may lend to the agent not diffusing in a concentric fashion, and that sets the stage definitely for some issues, maybe ruling something out as not very effective, and really it's the result of the medium. So what happens in these circumstances, we would think, if we're thinking a disease situation, we would have cultivated this bacterium from the patient. We would grow it up in some broth. We would pipette out a small amount of the broth onto one of these plates. Typically 50 to 100 microliters is added to the plate. We use a spread plating technique where we have the glass spreading rod spreading the bacterium all over the surface of the plate, creating what we call a lawn of growth. So the entire surface of the plate should have bacteria inoculated onto it. As soon as that broth has been spread out onto the plate, you give it a minute or two to kind of absorb into the medium, and then you apply the antibiotic discs. The dispensers we have a tamper that as soon as you press the dispenser The discs are placed onto the plate, and there's a little plunger that tamps each of the discs onto the plate. You flip it upside down and incubate it like you do with every other plate. After it incubates for 24 to 48 hours, depending upon the type of microbe, you pull the plates out and you can measure the zones of inhibition, potentially. As we look at Pseudomonas over on the real image, you can see here we've got one drug that's effective and 3 of them not. Pseudomonas aeruginosa is an organism, it's a gram-negative rod. It's an organism that is relatively antibiotic resistant. It does have some serious issues when we think about wounds, burn patients, things like that, where it may come in and colonize. It's also a good one, or a bad one, for patients with cystic fibrosis. It colonizes in the lungs and it becomes very, very difficult to treat because it is so highly resistant. It also forms biofilms. It's got a big capsule layer that sets the stage, all sorts of things that are kind of virulence factors. It's another one for ear infections as well, especially with little kids, and kind of recurring ear infections might be the result of Pseudomonas. Comparatively, we've got Staphylococcus on the other side, and I would imagine we've got the same 4 antibiotics on these 2 plates. And you can see there is definite more effect with the Staphylococcus versus the Pseudomonas. So I also, just one quick side note with this, as we see the 2 plates, you can see the Staphylococcus, kind of a white growth on these plates. And the Pseudomonas, these guys look green. And when I say a lawn of growth, Pseudomonas is the best one to really give you a true looking lawn, a nice green lawn. So kind of interesting. Pseudomonas produces a pigment called pyocyanin, which is this greenish pigment that the colonies exhibit. So you measure the zones. Here we've got just a handful of different antibiotics. It's not telling you which is which, doesn't matter at this point. But again, the bigger the zone, most likely the drug of choice. There are some that look as if maybe they're effective, but each drug has some specifics as far as the size of that zone. And just because it seems to be targeting it doesn't necessarily mean that that would be a drug of choice. There are some drugs that are more kind of just inhibitory and not actually, bactericidal, not killing. And so you might see a slight halo and then a little bit of pinpoint growth within that halo. That definitely indicates a drug that you would not want to prescribe. It's not going to be effective, all right? So this technique, disc diffusion or Kirby-Bauer technique, is a mechanism that is still utilized to figure out the specific drugs of choice. And in the next chapter, we'll mention a little bit of this. There are some antibiotics that are narrow spectrum, targeting gram-negatives or gram-positives, and there are others that are broad spectrum, kind of a kill-em-all sort of approach, where they will literally target any sort of microbe. That sets the stage for other issues with potential interactions with that and impacting your normal flora. So we'll hit that later as well. So again, disc diffusion, and we will see some similar techniques again in the next chapter, and taking it up a little bit more, we can do some dilutions and figure out specific concentrations, and that might be something that that we are interested in as well. So here, and this is, looking at disinfectant and seeing whether or not, it's effective or not. So we've got our disinfectant solution added into this broth with an inactivator, and then placing it onto the respective plates. Growth of a handful, 5 or more colonies, indicates contamination of the disinfectant solution. So typically, in the microbiology labs, you roll in there, you've got, some sort of dispenser, old soap bottle, that you squirt the disinfectant onto the bench with. In the stock room where they're preparing all of this, there's a 5-gallon jug where they prepare all of the disinfectant that goes out to all of the teaching labs, really all of the microbiology labs. You're not using disinfectant in an ecology lab, at least not typically. So there are questions, potentially there might be contamination within the disinfectant itself. And this is what actually is allowing you to see if that's the case. And if there is contamination within there, when you inactivate the disinfectant, growth will shine through. And that is really what we're kind of looking at with that kind of a quality control mechanism in this circumstance. So I think getting close to the end here, CRE is a drug-resistant strain. This is associated with nosocomial or hospital-acquired infections, and there are all sorts of possibilities with that, with procedures that may not be as clean or sterile as one might hope. So this duodenoscope has some issues where there might be some areas that are relatively difficult to disinfect. And in turn, there are definitely some potentials for infection to, microbes to be spread from one individual to another because of the inability to thoroughly disinfect the device. That makes you feel a little bit uneasy. At least it does me. So there are definitely, some potential issues when we think about some of this and the possibility of winding up with something else that you didn't bargain for. And that's definitely a problem. Let me stop sharing this and start sharing another one. Any questions while I'm getting this guy loaded up? Okay. You should be seeing chapter 14 now, I hope. So the antimicrobial drugs, this really, the last with the susceptibility plates is a pretty good segue into all of this, thinking about the whole gamut of different types of antimicrobial agents that we have access to. There are definitely issues when we think about, antibiotic resistance and, some super pathogens, those that are resistant to multiple classes of drugs, some of which maybe there isn't an antibiotic that is effective at targeting them anymore. And that is definitely grounds for concern. Okay, so as we start with this, there's a lot of, interesting history behind all of this. Penicillin, first antimicrobial that was discovered, and, we thought maybe this is or was some magic bullet. This is something that we could cure whatever ails you. And people were given shots of penicillin for anything and everything. It kind of seems as if that were the case. That sets the stage definitely for some issues. So here we can see some flyers from World War II touting penicillin, saying it saves soldiers' lives. And, it ultimately was credited for sure as, helping the Allies win the war. I think there were some other things that truly helped with that as well, but we're not teaching history here. So there's definitely, there were benefits, and through overuse and misuse, there's issues as well. So we do see, evolution of drug resistance with all sorts of different pathogens and all sorts of classes of antibiotics, not just penicillin. We do notice, in the early 40s when it really was kind of introduced in widespread use, it was discovered early 1930s, maybe mid 30s. I don't remember all of the numbers. Dates don't truly matter to me. So discovered, and then we'll see here in the next image a few others that ultimately bring it to mass production. Once we start seeing it in widespread use, shortly after, it was just a handful, 4 or 5 years after the first introduction, that we start recognizing some organisms are no longer targeted by it. There's resistance, and just prescribe something else. That is definitely a problem, and it's something that we're faced with currently, with the question, what do we do with this? There are all sorts of other potentials, and really depends on maybe where you go, where you might see other possibilities for millennia. Herbalists in China have used different types of plants for treating all sorts of different ailments, and some of which definitely harbor some antimicrobial properties. I have seen some stuff talking, ancient practices for different types of infections, and some of which, had been recorded, obviously, and tapping back into some of that stuff, or maybe some of this stuff right here, to see whether or not it holds some effect against these super pathogens, these that are highly resistant. And there's definitely some potential there with some of these substances to maybe be useful with these types of resistant type pathogens. So all sorts of things here that, I can't tell you what any of it is. I'm sure right here, the little card's probably telling you what's up, but we can't see any of that. Just a handful of different types of plants, herbs, whatever. So Paul Ehrlich, he was influential in discovery of a compound 606. It's an antimicrobial agent that was effective in treating syphilis. And I assume, this is, maybe multiple trials. It's like, there's all sorts of things, the WD-40, trial number 40. I think 409, the window cleaning stuff, I think that's a similar sort of thing. There's multiple attempts to get it correct. I think that's the idea with this. Trial and error. Science is not perfect. So interesting. And so one of the initial things that we think about, and again, I didn't go through and look into compound 606 to see the respective details as far as what that was. Fleming, right here, Alexander Fleming was the first to discover penicillin, and this was in the late 1920s, 1928. This was kind of, it was an accident, truly. So the story goes, he was studying, I want to say it was Staphylococcus aureus. I'm pretty sure it was Staph aureus. He was growing these bacteria up in the lab. Back in the day, you go to the lab today and there's all of these plastic petri dishes. Back in the day, they were all glass, and we would sterilize and reuse them. These glass dishes fit, kind of loosely, the lid loosely on the top of the plates. The issue was one of the plates, the lid was not fully on the plate. It was kind of just a little bit open, a little bit ajar. And it was over the course, maybe these plates were inoculated on the weekend, and he comes back a couple of days later. It was a couple of days of incubating where he comes back and sees this plate with the lid partially off, and there's a bunch of fungus growing in there, a bunch of contamination ultimately. And basically the first thought is, well, this plate's ruined. It's garbage. I need to just discard it. But he kind of took a look at it a little more closely and recognized where the fungus was growing, there was no bacteria. And there was, back to like that antibiotic plate that we saw, there was a kind of a zone, an area where the fungus wasn't. And then there was just empty space, if you will, a clear zone, and then some bacteria. And it made him kind of wonder, what is it that this fungus is doing that's preventing the bacteria from growing in the first place? So the organism was Penicillium, which ultimately, through purifying the specific substance, gives us penicillin. So kind of interesting with that. So he's first to actually discover that some of these fungi, Penicillium specifically in this case, has some antimicrobial properties. We flash forward a little bit. Flory and Chain discovered basically how to kind of industrialize it, if you will, how to scale up the production of penicillin, growing it in really kind of big vats to create mass quantities of this highly effective drug, right? They were able to purify it and show the effectiveness, the efficacy of it in animal and human trials in the early 1940s, which in turn set the stage to start widespread use. And again, one of the thoughts for helping soldiers in combat as they're treating all sorts of issues. Waxman, he was the first to show some other organisms, and I love seeing pictures of old lab pictures just to see all of the different types of tools and things that they utilized, some of which still potentially in use today. Pretty cool. This, I'm sure this flask has a name with this round bottom on it. So multiple ring stands holding it, the Bunsen burner underneath, purifying it, a little collection. It almost looks like we've got a type of still here where the bubbles, the gas will come up, it will condense, and we're collecting it down over here. So not that I'm trying to show you how to distill alcohol, but there you go. There would be a cooling tube through here where you'd have water piped through, and another tube of glass that is moving through where your solution's going through. So he found some bacteria, some soil bacteria, the actinomycetes. These are really kind of impressive organisms in the first place. And there are numerous types of antibiotics that we are able to isolate from specific types of actinomycetes. So usually we think about antibiotics, it's either some fungi or a few types of bacteria that have these capabilities. And the ideas really from an ecology standpoint would be to protect themselves. They're in the environment, growing, feeding, trying to, take in specific types of nutrients, and they want to keep that stuff for themselves. So by producing some sort of antimicrobial compound that they secrete outside of themselves, out into the environment, it helps to protect them by not allowing other microbes. It kills them off before they get in and rob them of their food. Kind of a brilliant strategy. So with that, antibiotics naturally occurring in some of these organisms, and with that said, antibiotic resistance evolved shortly after the production of antibiotics in these organisms. Some organisms develop strategies to still be able to come in and rob them of their nutrients, whatever it might be. So, drug resistance is not something new. It's been around for a very long time as well. It's just through our recognition of antibiotics and our widespread overuse and misuse that has lent to drug resistance on a broad scale, right? So, sidebar. Another interesting little tidbit with this, and again, there's lots of different types of actinomycetes. There are some of these guys that are, I think kind of interesting. There's lots of different organisms that have, you've seen some stuff in the lab, you've grown some stuff up, and you've probably smelled a little of it, and you're like, holy cow, that is terrible. That is nasty. And maybe you might think, well, that smells like butt, and because that's where it's coming from. All right, that's what I think. Some of these guys, they're soil bacteria, and you can grow up some colonies of these actinomycetes, and it smells like dirt. It smells very weird, and really, the odor of soil is the result of a lot of these microbes within there, the actinomycetes. You can smell it, if you garden and you're out tilling the soil, you can sometimes get this odor. If, there's just been a rain, and you can kind of smell that sort of earthy odor. We are in Idaho. If you've had the opportunity to be in a potato cellar ever, that smell, which smells like soil, is really the result of these microbes. That's pretty cool stuff. And if that doesn't make you want to be a microbiologist, I don't know what I'm doing here. All right. So I know it doesn't. It's fine. So when we think about this, and this is kind of thinking, back to that where I talked about the different types of antibiotics, there's broad and narrow spectrum antibiotics. In this case, we're looking at a broad spectrum type of drug. We're looking at a super infection. We're looking at maybe the potential for an overgrowth of one type of an organism. In this case, we're not talking about antibiotic resistance, but this one may have some resistance within it, all right? So there's other potential issues that might set the stage for resistance in some of these organisms as well. And we'll hit that here in just a second, all right? But we'll start off here. You've got your normal flora, normal microbiota. This is the good guys, the stuff you want. It helps to keep the bad guys in check, all right? The opportunistic pathogens, those that, given the right circumstances, can cause some serious issues, all right? So let's use Clostridium difficile as an example, an organism that you can find within your intestine in hopefully low levels. So you've got your good guys, you've got some bad guys. Some of these could maybe be some bad guys too, given the right circumstances, right? But in this case, this little guy right here, this dark one, is the one we're going to say is the pathogen, all right? All of these others, they're your normal flora. They're the good ones, all right? So you prescribe some broad spectrum antibiotic for some completely different ailment, all right? And ultimately, this broad spectrum antibiotic kills off the bulk of your normal flora. But this guy happens to have some resistance. It's not targeted. And as these guys, as their population is virtually wiped out, now all of a sudden there's all of this space. There's all of this prime real estate that's opened up for these guys to move into, to take over, to thrive. So the resistant populations, these pathogens now, spread, proliferate, and result in a super infection, where you've got basically the majority of all of the organisms are the bad guys. And that's going to require a completely different type of antimicrobial agent to try to target it. Narrow spectrum drugs are not going to target every single thing. It's maybe just these red guys that get targeted, leaving the other normal flora and leaving a lot of the real estate already purchased, if you will, so that the pathogen doesn't have that potential to proliferate. So deep down, if you're taking antimicrobial agents, you hope that it's a narrow spectrum and not a broad spectrum in the first place to help eliminate this possibility. And there are other, there are some procedures, some techniques, uh, you know, probiotics, eating yogurt when prescribed antibiotics helps to alleviate some of these potential issues. With that, yeast infections, Candida albicans is a fungal, it's a yeast cell, it's a fungal organism. So antibiotics targeting bacteria don't target the yeast cells. And it's the same sort of an idea where you have low levels of them growing as normal flora, but now wiping out the specific types of bacteria allows the yeast cells to proliferate, and similar sort of an idea as we see right here with the super infection. While we got this on here, I want to just talk briefly about the potential for drug resistance to ultimately show up within a population. And there's, disregard the top bit where it's talking about these things. You could have a handful of organisms, some of which are sensitive to the antibiotic, and some which are maybe marginally resistant to that antibiotic. Oftentimes, your pathogen might have some issues where it's maybe able to withstand, maybe able to deal with it a little bit better. There's numerous things that set the stage for issues with resistance. It could be, transferring genes from one organism to the next. Now, this one picks up resistance where it never had it before. Or it could be the misuse of antibiotics, taking something, but not taking the full course. Usually antibiotics prescribed for about 2 weeks. Oftentimes you're taking that antibiotic after about 5 days, you feel great, you quit taking them. That is terrible. Do not do that. And many people would say, oh, I've done that before. I felt better. Well, the issue is you've taken it, you've knocked down some of those potential pathogens a little bit to the point that you feel a little better, but maybe some of those pathogens are still there, they're still proliferating, and maybe some of those have a little bit of resistance within them. Taking the full course might knock their population down enough, maybe not completely eliminating them, but knock it down enough that you get to this situation where there's a lot of the good guys and very few of the bad guys, and everything's relatively fine, right? But given that circumstance, only taking a partial dose creates a bigger population of these bad guys. And so now instead of just one of them, maybe you've got, half of this image right here are the bad guys. And that sets the stage for them to replicate. And then you go back, after that infection didn't really seem to clear up, and you start eating the rest of your antibiotics to find that it's not effective. You go back to the doctor, they prescribe you something else, and you do the same thing, not telling them that you didn't take the drugs the way you were supposed to. And that sets the stage for really the formation of drug resistance, as you've misused those antibiotics by not following the directions. And that has nothing to do with the overuse or anything else like that. Multiple issues with that. So take home message, if you're prescribed antibiotics, take the full course. Even if you start to feel better, make sure that you take the entire course of the antibiotics, and don't share your antibiotics with people. Don't save them and share them. They are for you. All right. And again, if you've saved them, then you haven't taken the full course. That's an issue. So here we've got a couple of different potentials as far as how you may be taking these antibiotics. And oftentimes we think oral administration, but it could be intramuscular or intravenous. And some of the drugs are really pretty potent, pretty dangerous. Oral medication is not warranted. It might require IV administration. And there are even some where they directly inject it into the heart, which you're in bad shape if that's happening. So here, We've got a couple of different things on here. Plasma concentration on the y-axis, time, and it's all arbitrary right here, but time on the x-axis, T0 or T0 is when you're given that drug, when it's first administered. And then we're looking at really how quickly it gets through the body, really thinking about getting to the source in the first place. So IV administration, you see it spikes very rapidly, and then we see it declining. They all decline over time. And again, thinking, maybe repeated administration, keeping these levels elevated for prolonged periods of time, definitely important. This might be weeks as we're looking at it. I don't think it actually tells me in this when I looked at it before. It matters not, like I said, it's arbitrary. So the intramuscular, you can see it's injected in, and we've got relatively high levels early on, early on, and then peaking, and again, declining down. The oral administration definitely takes more time. It's got to get in to the body and move throughout, hopefully reaching it in a relatively quick amount of time, but it's definitely slower, right? And then going down, all right? So multiple potentials as far as how it reaches the target area and the time needed for that. So with that, I think that's probably the best. Well, let me hit this real quick and then we will stop. So this is kind of interesting to me. It's a little bit disturbing. This is parental expectation of antibiotic use from 2000, and then another survey, 2013. We're looking at Medicaid insured and commercially insured individuals, and the thoughts, the expectations, and they are definitely varied between the types of insurance. And I think that's a huge concern in the first place. So the idea with this, trends as far as expectations. Do you think you should have it or not? And what, and it really kind of begs the question with this. So are there discrepancies? It seems as if that's the case. So if my doctor does not prescribe an antibiotic when I think one's needed, I'll take my child to another doctor. And you can see all of the Medicare it all's like this, right? So it's more likely that they will do that. So here it was 10% in 2013, 25% would say that. With the commercially insured, it's roughly the same at about 10% across the board. If I expect an antibiotic, I am less satisfied if I don't receive it. About 15% with the commercially insured from 2000 to 2013. It's relatively steady. They increase a little, but it's relatively steady. With the Medicaid, 10% in 2000, upwards of almost 25% in 2013. Here, the last one, I'd rather give my child an antibiotic that may not be needed than wait to see if she gets better without it. And the commercially insured, has declined, so which is good. So that less people thinking that that's what should happen. And truly, you should not be prescribing antibiotics if they don't need it. With resistance is an issue for this fact. So I'm going to leave it at that. There's definitely some issues with that, and I think proper education is truly what's needed to ensure that people don't just think I should take antibiotics. I went to the doctor and I want an antibiotic. That's not what's good for anybody. Okay, enough of that. I will quit. Any questions before I leave? If not, I hope you all have a good weekend. I will see you in person on Monday for those of you that will be attending. Otherwise, lecture will be posted. Looks like I've got one chat. Let me look at it real quick. Oh, thank you. You are welcome.
Thank you.
Thank you. Have a good day.
You too.
Okay.